Welcome everyone. In the unit, we are going to study metabolism of lipids. Lipids mainly include the fat, such as triglycerides, fatty acids, and cholesterol. Related molecules in the body, lipids are major source of long-time energy, and they also help in insulation and cell structure. In this lesson, we will understand how what is fat is broken down is least energy and what happens when lipid metabolism becomes abnormal. This unit focuses on metabolism of lipid. We will learn to lipolysis, which means breakdown of stored fat into fatty acids. Then we will study beta oxidation, which is the main method by which fatty acids are converted into energy inside the mitochondria. After that, we will learn ketogenesis and ketolysis, which is explained by ketone body formation and utilization. Finally, we will look at disease caused by abnormal metabolism such as ketoacidosis, fatty acids, and hypercholesterolemia. Lipid contribute around 50 20% of total body weight in human. The major stored from lipid and our body triglyceride, also called triglyceride, and is stored mainly adipose tissue. The stored fat act as a big energy reservoir, especially during the fasting or long starvation. Fat is very energy rich because fatty acids are in a highly reduced form, so they release a large amount of energy when oxidized. Fat also act like a insulating layer and help maintain body temperature. Lipid acid, different form like triglyceride, phospholipid and fatty acids. By triglyceride form 85% 90% of total body lipid. This breakdown of stored lipid release fatty acids and called lipolysis, and this become important during long fasting when carbohydrate store is reduced. This slide shows as a overall flow the lipid breakdown mainly from stored triglyceride to the fatty acids and glycerol. The idea is simple: stored fat does not directly give energy until it is broken down into the fatty acids. These fatty acids then transported to tissue like muscles and liver where they are oxidized to produce ATP. So, at long fasting, the body slowly shifts from using glucose to using fatty acids as a major fuel. The slide shows DG, which was stored in adipose tissue, is broken down step by step into free fatty acids and glycerol. This breakdown is done by enzyme called lipase, which energy needed TG are broken and fatty acids are released into blood. The fatty acids travel with albumin and plasma and release to this is starting point lipid metabolism during fasting and starvation. This slide explains DG lipase regulation, meaning how lipolysis is controlled. DG lipase activity is mainly controlled by hormones during fasting and stress. Hormones like glucagon and adrenaline activate lipolysis, so fatty acids released for energy. After meals, insulin reduces lipolysis and promotes fat storage. So the body uses hormones like gastric insulin as a store fat and glucagon adrenaline say break fat and use it. This adipose release by the bacteria and glycerol during lipolysis, but adipose to leg an enzyme called glycerol kinase. Because of that glycerol cannot be usually efficiently inside the adipose tissue itself. So glycerol is transported to liver where it is converted to glycerol 3 phosphate and used to gluconeogenesis other pathway. In contrast to fatty acids released from adipose tissue go to tissue for energy production through beta oxidation. This is the way fasting liver becomes a major organ controlling by glucose and lipid metabolism. Now we begin the most important energy pathway for fat called beta oxidation. Beta oxidation means the fat is the kind is broken down step by step inside the mitochondria to form acetyl-CoA. The acetyl-CoA then enter the TCA cycle or form ketone body depending on the situation. So beta oxidation is central pathway by which becomes the energy. Athletics are a major energy source in tissue with higher energy demand such as the heart, muscles and liver. During that fasting, fatty acid oxidation become more active because glucose supply become limited. When the body oxidation fatty acids produce large amount of ATP compared to glucose. This is why fat is concentrated to best long-time energy store. But due to acidosis fatty acids, the body must first activity them to transport them into the mitochondria. Now we study that this will step by clearly. The first step the beta oxidation activation by fatty acids into the acetyl-CoA. This activation is done by the enzyme cyclone kinase, also called acetyl-CoA synthetic. Without activation, fatty acids cannot enter the oxidation cycle. This activation step uses ATP, so it is like preparing the fatty acids and breakdown. Now we understand fatty acid activation more clearly. Fatty acid activated to acetyl-CoA by thiokinase usually into outer mitochondrial membrane or cytosol. In this step, ATP is used and converted to into AMP and pyrophosphate. So it is consuming energy the activated product. Alkyl coenzyme is now ready for transport into mitochondria. This step is essential because it committed is fatty acids towards oxidation and energy production. Now we move the testing stage fatty acids oxidation inside the mitochondria. Even though fatty acids are activated, they still cannot directly enter the mitochondria matrix easily. So the body uses a transport system called in the carnitine successively. After the transport, the fatty acid undergoes repeat cycle to beta oxidation and produce acetyl coenzyme. This slide explains an important point. The inner mitochondrial membrane is impermeably for fatty acids actual coil. That means fatty acids cannot cross into mitochondrial matrix the seamlessly. So body usually creatine and carrier. The first fatty acid coil component carnitine from acetyl carnitine. The cycle carnitine crosses inner mitochondrial membrane. Then inside matrix is converted back to acetyl CoA. Carnitine return to outside to bring another fatty acid. This subtle system important for long chain fatty acid. This slide usually shows the carnitine physical diagram. The idea to remember is CP2-1 from actual carotid the transport move across and CP2 reform alkyl CoA inside the matrix. The carotid low or the utility refers to fatty acid concentration reduce and energy production become more stable during the fasting. Now we study the beta oxidation proper which happens in the mitochondrial matrix. Beta oxidation occurs to a repeating cycle is a cycle is shorten the fatty acid chain by 2 carbon and produce 1 actual CoA during this cycle. NTH and FCATH2 really also produce which are later generated ATP in cell electron transport chain. So beta oxidation produces energy in two ways. Directly by forming actual CoA and indirectly by forming NATH and FATH2. This slide usually shows the 4 repeating reactions for beta oxidation, oxidation, hydrogen oxidation, and thiolysis. The main point of the 4 steps repeated again one until the entire fatty acid is converted into the alkyl CoA unit. This is where is called a separate pathway. Once alkyl CoA is formed, it can enter the TCA cycle to produce more energy. This slide typically explains the palmitic oxidation as a sample. Palmitic acid is a 16 carbon fatty acid during beta oxidation. It is produces multiple alkyl CoA molecule. Cycle to 2 carbon so palmitate produce 8 alkyl CoA after completed breakdown. It also produces NTAH and FCATH2 repeatedly which produce ATP. Example is important because enzymes often react beta oxidation to palmitic acid ATP yield. Now we start ketogenesis. Ketogenesis is formation keto bodies in the liver. They happen mainly when the body is fatty starvation and uncontrolled diabetes or very low carbon intake when fatty acid oxidation produced to much actual CoA and the TCA cycle cannot handle all of it. The liver converted actual CoA into the ketone body. This ketone bodies can be used as an alternative fuel mainly to escape during prolonged fasting. This slide shows the reaction to ketogenesis frequency. In liver mitochondria actual CoA will combine to and form intermediate like actual CoA and HMC CoA and they finally produce ketone body. The main ketone body is acetoacetate, beta-hydroxybutyrate, and acetone. The acetone is volatile and can escalate. We see where fruitly break may occur during the ketose. The important point in ketone body is produced mainly by the liver, but the liver itself does not use that much. Is exported in other tissue. This slide usually shows the diagram of ketone body formation and their movement to other tissue. The key idea that ketone body are water soluble, so travel is easily into blood. They are used by heart muscles and during prolonged fasting. The brain also starts using the ketone bodies. This helps reduce the need the glucose and productive body protein form begin broken down too quickly. This slide introduction to ketogenic diet. Yeah, ketogenic diet is a high fat, low carbohydrate diet that promote to ketone body formation. When carbohydrate intake is low, insulin decrease and the fat breakdown increase. So ketone bodies become higher. The ketone diet have a medical usage. It Some cases, such as drug agitation, escapably under medical supervision, but uncontrolled glucose can use dangerous diabetes, leading to ketoacidosis. This slide explains when the ketone body increases too much. Ketone body production increases, particularly into uncontrolled diabetes, prolonged fasting, starvation, and sometimes during the low carb diet. When ketone bodies are overproduced, blood becomes acidic because ketone bodies are acidic in nature. It can lead ketoacidosis, which is a medical emergency. So ketone bodies are usually useful, but excess production becomes harmful. Now we learn to ketolysis, which is the main relation by ketone body by tissue. Ketolysis occurs mainly at hepatic tissue, such as heart and muscles, and during prolonged fasting. The brain also uses ketone bodies. In ketolysis, ketone bodies are converted back into acetyl-CoA, and then acetyl-CoA enter the TCA cycle to produce ATP. This is how ketone bodies provide energy. The liver produces ketone bodies, but other tissue consume them. Now we start this is related to abnormal lipid metabolism. When lipid metabolism is not balanced, fat may accumulation lipid levels in blood and may become abnormal. It can cause important disorder like ketoacidosis, fatty acids, and hypercholesterolemia. This disorder clinical important and also accident to the exams. The slide explains diabetes relation with lipid metabolism and uncontrolled diabetes influence shallow effective so lipolysis increases greatly. Main lymphatic reach the liver undergo beta oxidation. This produces large amount of acetyl-CoA and the ketone bodies increase. As a result, the patient may be develop ketoacidosis also. Triglyceride and cholesterol level may be reach the blood, increase the risk of complication. Finally, this slide explains the fatty acids. Also, hepatic steatosis normally in liver contains around 5% fat, and it is not a fat source organ like adipose tissue, but it is a condition tissue accumulated in the liver and causes fatty acids. This can happen due to alcoholism, uncontrolled diabetes, starvation, obesity, or impaired lipoprotein formation. Fatty acids can cause liver enlargement and may process to liver inflammation and damage if the causes continues.